One of the cooler labs we do in this class is to show antibiotic production from soil bacteria, and it goes a long way towards demonstrating the role of antibiotics, where they come from in the natural environment. So this experiment, if we haven't done it in class already, involves taking a sample of dirt and a test tube, and this is a petri dish here, an empty petri dish. Combine the dirt, diluted dirt, with some agar, the solidifying agent, and then you take that and you pour it into your plate, like that. All right, now what you have is a plate of agar with some diluted dirt in it, and these dots are meant to represent, there's a lot of different cells in the agar, some of which will grow, some of which will not. But if we focus down in on these cells, here we have 3 different cells. Okay, I've actually drawn them bigger. This is not to scale. We have 3 different bacterial cells here. If we let time go by, okay, the red colony is getting bigger. Okay, it is also producing antibiotic in this case. Now this is where diffusion comes in. When you learned diffusion in 1151, you talked about diffusion in 1611 and 12. This is happening on this plate, right? Because where another bacterial cell is in relation to this giant antibiotic producing colony is going to dictate whether or not it can grow. If it's close enough to that antibiotic producing colony, it's not going to grow. Whereas if it's further out away from the colony, it's going to be able to grow. So as we let time go by then on this, okay, we pull out a little bit. Here's our petri dish, and we just let that incubate overnight. These colonies that are further away will grow, and they'll form this hole, a zone of clearance there. And that zone of clearance, why cells are not growing in that area around that red colony, is because the black bacteria is sensitive to whatever antibiotic is being made there. That is the most direct demonstration I can think of to show that antibiotics basically come from microbes in the natural environment.